Assalamualaikum and hi everyone. Our group will present Experiment 5, Gas Chromatography or GC. Gas chromatography or GC is an analytical technique used to separate the components of a mixture based on their volatilities. It is especially suitable for compounds that can be vaporized without decomposing. In gas chromatography, the sample is first injected into the instrument and vaporized. The vaporized components are then separated because they are partitioned between two phases, a mobile gases phase and a stationary phase. The stationary phase may be a liquid or a solid material held inside the column. The mobile gases phase is known as the carrier gas. Common carrier gas used in gas chromatography include helium and nitrogen. The carrier gas transports the vaporized sample through the column. As the sample moves through the column, each component interacts differently with stationary phase. Components with higher volatility or weaker attraction to the stationary phase move through the column more quickly and elute earlier. Components with lower volatility or stronger attraction to the stationary phase remain in the column for a longer time and elute later. The flow of carrier gas through the column is produced by pressure from a compressed gas cylinder. This continuous flow carries the separated components to the detector where a chromatogram is produced. Then the chromatogram can used to determine the retention time and identify the component present in the sample.
I will explain the principle of gas chromatography. Gas chromatography is an analytical technique used to separate and identify volatile compounds in a mixture. The separation occurs because different compounds travel through the column at different speeds. First, the liquid sample is injected into the instrument and immediately vaporized. Next, an inert carrier gas such as helium and nitrogen carries the vapor through the chromatographic column. Inside the column, the compound interact differently with the stationary phase. Compounds that interact less strongly move faster and leave the column early, while the compounds that interact more strongly take a longer time to pass through the column. As each compound exits the column, it is detected and recorded as a peak on the chromatogram. Therefore, each peak on the chromatogram represents as a different compound, and its position gives the retention time of the compounds.
At the end of the experiment, we should be able to determine the retention time TR of N-butanol and 2-propanol, identify the components present in a standard mixture based on the TR, identify the component present in an unknown sample, and determine the effect of temperature on TR and RS.
This slide shows the main component of a gas chromatograph and their function. First is the carrier gas cylinder, which supplies an inert gas that transports the sample through the instrument. Second is the pressure regulator or flow controller, which controls the pressure and flow rate of the carrier gas. Third is the injector, where the liquid sample is introduced and vaporized. The sample then enters the column inside the oven. This is the most important part because the separation of compounds occurs here. The oven controls the temperature of the column. After leaving the column, the compounds reach the detector, which detects each compound as it in use. Finally, the signal is sent to the computer and decoder, which produces the chromatogram and calculates the retention time and peak errors. All these components work together to separate and identify the compounds present in the sample.
This slide shows the main component of a gas chromatography and their function. First, the carrier gas cylinder supplies an inert gas, such as helium or nitrogen. Its function is to carry the vaporized sample through the column. Next, the pressure regulator and flow controller control the pressure and flow rate of the carrier gas. This ensures that the sample moves through the instrument at a consistent rate. The injector port is where the liquid sample is introduced into the instrument. The injector is heated so that the sample is rapidly vaporized before entering the column. The column is the main part of GC instrument where separation occurs. It contains the stationary phase. Different compounds travel through the column at different speeds depending on their volatility and their interaction with the stationary phase. The oven surrounds the column and controls its temperature. The oven temperature affects the retention time and resolution of the peaks. A higher oven temperature generally causes the compounds to elute faster, while a lower temperature causes them to remain longer in the column. After leaving the column, the compounds enter the detector. The detector detects the separated compounds and converts their presence into an electrical signal. And finally, the computer or data system records the detector signal and displays it as a chromatogram. It is used to obtain information such as retention time, peak area, peak height, and peak resolution.
Now I will explain the experimental methodology used in this gas chromatography experiment. First, the gas chromatograph was set up with an initial oven temperature of 70 degrees Celsius, while both the injector and detector temperature were maintained at 180 degrees Celsius. Next, each pure component, which was 2-propanol and N-butanol, was injected individually into the GC instrument. The retention time of each compound was then recorded. After that, a standard mixture containing both compounds was injected. The peak obtained were identified by comparing their retention time with those of the pure components. Then, an unknown sample was injected into the instrument. The compounds present in the unknown sample were identified by comparing its retention time with the retention time obtained from the standard mixture. To study the effect of temperature, the oven temperature will increase from 70 degrees Celsius to 100 degrees Celsius, and the mixture was injected again. The change in retention time and resolution were observed. Finally, the oven temperature was further increased to 140 degrees Celsius, and the standard mixture was analyzed once more to determine how increasing temperature affect the retention time and the separation of the compounds. These steps allow us to identify the compounds present and investigate the effect of temperature on gas chromatography separation.
On this slide, the chromatograms of pure components at an oven temperature of 70 degrees Celsius are shown. The pure components were injected individually to obtain their reference retention time. For 2-propanol, one mean peak was observed at retention time of 2.707 minutes. For N-butanol, one mean peak was observed at a retention time of 5.955 minutes. These values were used as reference retention times for the identification of compounds in the standard mixture and unknown sample. Since 2-propanol had a shorter retention time, it eluted earlier than N-butanol. This indicates that under the same GC conditions, 2-propanol was more volatile and interacted less strongly with stationary phase than N-butanol. On this slide, the chromatograms of standard mixture at 70 degrees Celsius, 100 degrees Celsius, and 140 degrees Celsius are presented. At 70 degrees Celsius, two mean peaks were observed. Peak 1 had a retention time of 2.674 minutes, which was close to the retention time of pure 2-propanol at 2.707 minutes. Therefore, peak 1 was identified as 2-propanol. Peak 2 had a retention time of 5.814 minutes, which was close to the retention time of pure N-butanol at 5.955 minutes. Therefore, peak 2 was identified as N-butanol. When the oven temperature was increased to 100 degrees Celsius, the retention time decreased to 2.274 minutes for peak 1 and 3.281 minutes for peak 2. At 140 degrees Celsius, the retention times decreased further to 1.464 minutes and 2.248 minutes respectively. This shows that increasing the oven temperature causes both compounds to elute faster. At a higher temperature, the compounds spend less time interacting with the stationary phase, so their retention time becomes shorter. However, the peaks also become close together at higher temperature, which may reduce the peak resolution. Therefore, lower temperature give a longer analysis time but better separation, while higher temperature give a faster analysis but potentially poorer resolution.
Now I will discuss the chromatogram of the unknown sample. The chromatogram shows only one peak with a retention time of the 5.957 minutes. When this value is compared with the retention time of the pure compounds, we can see that it is almost identical to the retention time of pure N-butanol, which is 5.955 minutes. The difference is only 0.002 minutes, which is very small and can be contributed to minor experimental error. Therefore, we can conclude that unknown sample contains only one compound, which is N-butanol. This experiment also investigates the effect of temperature on gas chromatograph. At 70 degrees Celsius, the peaks appear at longer retention time because the compounds move more slowly through the column. However, the separation between the peaks was very good. When the temperature was increased to 100 degrees Celsius, both compounds elute faster and the retention time decreased while the separation remains acceptable. At 140 degrees Celsius, the peaks appear much earlier and become closer together. This indicates that the resolution decreased. This happened because increasing the temperature increased the volatility of the compound, allowing them to move through the column more quickly and spend less time interacting with the stationary phase. Therefore, increasing the temperature makes the analysis faster. But if the temperature becomes too high, the quality of separation decreases and peak overlap may occur. Based on chromatograms obtained, the temperature has significant effect on both retention time and peak resolution. At lower temperature, compound remain in the column longer and separated more effectively. As the temperature decreases, the retention time decreases because the compound vaporizes more easily and move faster through the column. However, the resolution between the peaks also decreases because the compound spend less time interacting with the stationary phase. In addition, the chromatogram of the unknown sample confirmed that the unknown compound was N-butanol because its retention time closely matched that of pure N-butanol. In conclusion, the retention time of pure 2-propanol and N-butanol was successfully determined to be 2.707 minutes and 5.955 minutes respectively. The standard mixture was identified as containing 2-propanol and N-butanol, while the unknown sample was identified as N-butanol. The experiment also demonstrated that increasing the column temperature decreased the retention time, but also reduced the resolution between peaks. Therefore, gas chromatography is an effectively analytical technique for separating and identifying volatile compounds based on their retention times and chromatographic behavior. Thank you.